Hey everyone, this is our lesson on enzymes and metabolism. So by the end of this lesson, you should be able to explain how enzymes act as catalysts in biological reactions. So first, let's make sure we understand what metabolism is. I know you've heard that term before, so let's just make sure we know what that means. So metabolism is the set of chemical reactions that occur in living organisms. And this has to be done to maintain life in that living organism. So it's learning how to make and use energy, really. So this process, or these processes as they occur, allow your organisms to grow and reproduce, maintain their structures, and respond to their environments. So you can see in this little GIF I have over here, the sample that's like 2DG, I think is what it's called. It actually spreads out faster, which means it has a faster metabolism. It's able to use that source quicker. So now that we know what metabolism is and we know that it deals to the chemical reactions within the body, let's talk about those a little bit. So chemical reactions absorb or release energy based on what they're made up of and what's happening. So in a chemical reaction, we see that new substances are produced by breaking or forming chemical bonds. So you're going to take your original compounds, break the bonds in them, and then make new bonds to form your new compounds. So first we have reactants, and reactants are substances that are combined or broken apart. So these are the ones that we start with because they react. Then we have products, and the products are the new substances that formed because they're produced. So we didn't go very crazy with how we named these. So we take our reactants, we add them together, and they will form our products.
That's crazy.
It is crazy. So now we have parts of a chemical reaction.
That pink stuff just exploded out of that cup.
It did, didn't it?
What was it?
Elephant toothpaste. So starting with chemical reactions, when we start them, we have to have some amount of energy to make the reaction take place. This is known as our activation energy. So it's defined as the extra energy required to break the bonds in your reactants to start a chemical reaction. So if you look at this GIF over here, we've got our reactants. They have to gain energy to get to the top of the hill so that they can complete the reaction. I like to say this is like a roller coaster because you have to always go up that first big hill before you can go through the rest of the ride because you've got to build up your momentum and your speed. Well, that's what a chemical reaction does. It's got to build up energy to make it through the rest of the reaction. But we can sometimes see that reactions are very hard to occur because the energy needed to start is so high. Well, to prevent that, we have what are called catalysts. And catalysts are substances that will speed up chemical reactions, but they remain unchanged themselves. So they are not a reactant or a product. They are just something in there to help speed things up. Metabolisms could not function without catalysts because they allow organisms to drive reactions. So what a catalyst does is it allows a different pathway for a reaction to take place that uses less energy. It needs less energy than the original reaction did. And then in our body, our biological catalysts are enzymes. And they allow reactions to proceed quickly and efficiently in the body to break things down. They work by lowering the amount of energy needed for the chemical reaction to occur. So that's what enzymes do. And we'll look at those right now. So enzymes are proteins. So remember, these are proteins. And they are catalysts that live in the body. So we define these as biological catalysts. What an enzyme does is it attaches to a substrate, which is the substance that is changed or acted on by the enzyme. So the enzyme itself would be considered a reactant. The substrate is another reactant because it's what is being changed. And the substrate is changed into whatever your end products are. So it is manipulated by the enzyme and changed. It does this by attaching to the active site of the enzyme. So this is where your substrate will bind to. And this is what lowers the activation energy. But keep in mind, active sites are very peculiar. It's like a puzzle piece. Only certain substrates will fit into that active site. Other ones will not, which means that enzyme will not work on them. Okay. So we do have to pay attention to that. And sometimes if you have overactive enzymes, you can add in inhibitors, which will lock into that substrate, that active site, and keep other substrates from being able to attach. All kinds of different things here. But the active site is where the reaction takes place. And then most of our names for enzymes end in the ACE ending. So protease, lipase, helicase, things like that. So those are enzymes. And then they bind, like I said, to specific substrates because of their shapes at the active sites. A denatured enzyme means that the shape of the active site or the enzyme itself has changed because the changes in pH or temperature. And when this happens, your enzyme does not work anymore. So it will keep your substrates from binding, which means they become ineffective. These are very pH and temperature sensitive. So the enzymes in your mouth would become denatured if they move to your stomach because the pH is different. Sometimes you'll denature enzymes when you get a fever and you have a fever for a long time. That's why it takes a long time for